Thank you, Morgan, for the introduction. Okay, I'm assuming everyone can see my screen properly. So today I'll be talking a little bit about what's going on under the hood when we talk about whole genome characterization of SARS-CoV-2 samples for public repositories. I think as many of the audience know, there's been a lot of activity within OICR supporting various initiatives around the province over the last year. So I wanted to tell a little bit about the story of how we got to where we are, and then finish off really kind of giving some insight into what has actually gone into these assays. And I know many appreciate that it's not just sequencing samples, there's a lot to consider, and I wanted to share a little bit about that today. So my group is the Genome Research Platform within the wider genomics program at OICR and UHN. My group is downstream from OICR's Tissue Portal group, which handles sample intake, and upstream of genome sequence informatics, which is led by Morgan. So the whole program primarily focuses on cancer genomics, basically starting from incoming tissues, ending with clinical reports produced by the Translational Genomics Lab, and like Morgan alluded to, under a CAP accreditation pipeline and standards that go with that. But to support research, our cancer pipeline can be broken down into a series of on and off ramps, where people can come in with any form of samples or analytes or libraries that they'd like to sequence. And there's a wide variety of outgoing data deliverables that we can do. Now, normally that whole process takes about 45 days for cancer genomes, sometimes more, sometimes less. The big challenge that we've had over the last year was how to squeeze that timeline down to as little as 48 hours for COVID genomes. So it's not just condensing all the components. We really had to rethink how we were doing certain things to customize an assay to support all of the viral activity that people have been interested in. So over the last 18 months, this curve might look quite familiar. It's the case count, weekly case counts for Ontario. We all started in this together in March of 2020. And back then, nobody really knew exactly how high this curve was going to go. And we were kind of told to go home, but go home and close the lab, but plan for 10,000 samples per day. So the next month, there was a lot of spaghetti thrown at the wall. A lot of methods were developed across the program in my group, ranging from various amplicon-based library preps, hybrid captures, straightforward RNA sequencing and data analysis to look for viral genomes. And eventually, the amplicon-based approach seemed to stabilize and standardize. And then later on in the fall, after we had been running amplicon sequencing for various hospital collaborators and a national network, CancoGen, we started collaborating with Public Health Ontario, which is at the Mars Center. And that was quite fruitful as the second wave rose. And in December at the peak, we were asked by various leaders around the Discovery District to plan for 10,000 samples a week. And I'll go into a little bit about that. Thankfully, we never got to that point, but I think it's worth kind of showing a little bit about what went into rethinking how to possibly do that if it came to be. So as I started saying, most of our work has been supporting global databases with whole genome sequencing of viral samples. We've sequenced a little over 4,000 whole viral genomes to date for various purposes. Some resolutions of hospital outbreaks, a lot of variant of concern confirmation and tracking to support epidemiology with Public Health Ontario. But the most has been for public databases, like GISAID here in the middle. We're part of the CancoGen virus sequencing initiative at the Genome Canada level and the OnCoV Genomics Coalition at the provincial level. And we've been working with various hospital groups as shown below. One thing I do want to also mention is that because we've been collaborating with Public Health Ontario, OICR was actually the first site to identify B1351 in January We confirmed that for our partners, and we also were instrumental in confirming B1617 in Ontario as well. So we've been pretty much right at the front line of all these activities, and been able to produce lots of timely data for the province. So the remainder of my talk will start going a little bit into the weeds. The general sequencing pipeline is probably familiar to many people. We start with receipt of RNA or samples, which is noted here from an upstream partner. There's a cDNA conversion step, and from that we can either choose to follow an Illumina library prep or a nanopore library prep. Most of the volume has been on the Illumina platform. Following raw sequencing, there's some primary sequence analysis and alignment by the genome sequence informatics team, and then secondary reports are primarily done by Jared Simpson's groups at OICR. As you can see, a lot of people have touched on this over the last year, and obviously not everyone is needed for every single run and plate coming through, but it has actually spread and involved a lot of people across the program, so it's actually been quite exciting. The one little note about the secondary reports from the Simpson lab, one of the key things in that bioinformatic pipeline is the output of these kind of phylogenetic heat maps that not only take each strain and characterize it in terms of what strain label it belongs to based on the variants it contains, but then you also get the relatedness about different samples. And so this is just a snapshot of a random plate of positive samples that we've sequenced for variant of concern screening. But if this was a plate, let's say from an outbreak, you could very quickly discover which samples were highly related to others simply by looking at the tree, and then maybe deducing that there's some kind of transmission going on in a hospital or a clinic, or if it's just chance encounters of multiple people arriving at the hospital in a short amount of time, but with completely different strains. So this kind of molecular contact tracing information is very useful to resolve those kind of outbreak questions. And I believe it's only can be done by whole genome sequencing. Okay, so going a little deeper into the library prep process, this is how the mplicon panel pretty much works. So the first step after you have viral RNA, you do a reverse transcription followed by two-pool PCR with a panel of amplicons designed by the Arctic Viral Tracking Network. It's a well-known international group that has done similar things for SARS and Ebola tracking. And so it's a series of overlapping amplicons split into two pools, shown here in black on the upper right figure. So that the top series of black amplicons would be in one pool and the bottom ones are another. That'll be a little relevant in a later slide. But they're done separately, and after the products are cleaned up and quantified, they both get pooled and put into an Illumina NextEraFlex library prep. Libraries are cleaned up and quantified, and then there's pooling and sequencing. So basically, this is a rapid way of reducing an RNA sample to raw coverage, shown here across the SARS-CoV-2 sequence. There's obviously some variability because there's variability on each of the amplicon performances, but all in all, you can see that usually if everything goes well, the whole genome is covered quite deeply, and certainly enough to call variants across a sample that's essentially haploid. For those that are interested, at OICR, we kind of separate the two activities, both in pre-PCR and post-PCR activities. The handling of RNA is done in a BSC. Pre-PCR areas are used for master mix preparations, and then once it goes to an actual Arctic PCR, it goes into the post-PCR lab. The rest of the library prep occurs in the post-PCR lab and usually just takes up one EP motion station. So if any of you have heard about robotics being used, they can be as large as full-fledged walkaway robots, but we've actually found that the EP motion instrument here by Eppendorf is basically a 96 head multi-channel, and it actually works quite well and without the sophistication of robots. Okay, so once we had an assay working, we wanted to obviously see how well it could detect dilute samples. We did a basic titration experiment to show that our implementation of this RT-PCR-based sequencing test detects down to 2,000 copies of RNA per mil. That corresponds to CT values of 37 to 42, which are extremely low. Typical positive samples are less than 30 and usually in the 20 to 25 CT range. One thing I do want to impress on you is that detection by sequencing does not automatically mean whole genome sequencing. So because we're interested in the research application of full data for as much of the viral genome as possible, we could see from this experiment that by using equal amounts of reads to sequence each sample, going from CTs of 21 to 41 at basically the limit of detection. The reason for this curve is that as you go to more dilute samples, more of your library is actually mapping to either something else or not mapping at all. So what we're measuring here is that at reasonable CT values, most of the library's reads belong to the SARS genome and are amplified properly, the remaining being adapter dimers or contamination or other odd products. And then as you go to a more dilute sample, it just becomes more difficult to generate a library with consistently high amounts of viral RNA inserts. This actually does compare favorably with RT-PCR-based detection. So the figure here on the left is showing the probability of a positive test for various modalities. This is from one of the recent CDC Science Updates, still adapted from further adapted from a paper. And you can see that as you go to different methods of verifying whether an isolate has virus in it, you need larger viral loads to infect vero cells, shown in the green and purple plots. And if you're using an antigen test, you need slightly less than actually infecting live cells. Probably, if you get a 50% probability at around 50,000 copies per milliliter of virus. But RT-PCR has a 50-50 chance of being successful with as little as about 200 copies per mil. So what we're seeing with the whole genome assay, given that it's the same basic technology plus a library prep on the end, is actually in the range of success for this kind of a diagnostic assay that's based on RT-PCR. The other thing that I wanted to note is that we're using a fairly modular assay design. It would have permitted scale up had it come to pass. And like I mentioned a few minutes ago, we were asked to plan for 10,000 samples per week. This was actually right before holidays. We were thinking about how would we do this either over the holiday or in January. We came up with a very compelling model of how we could utilize different teams to shuffle many plates around every day to basically try and push through as many samples as possible through our sequencing infrastructure. We figured we could probably do about 800 samples every day, which wouldn't get you to 10,000 per week, but would get you a lot closer to that number than we were at the time. Like I said, we had a whole plan. It didn't come to pass because the second wave started to decrease with the Ontario lockdowns. But it was actually a very interesting exercise for us to really try to squeeze out as much efficiency out of our lab processes as possible. Okay, so going back to the assay, we're basically running all these samples in a plate-based format, and each plate can contain up to 7 controls in a couple of groups, depending on the application. So we can have 2 positives for RT-PCR and library preparation. There's an additional 2 negative controls for extraction, which are basically either water or ideally viral transport media with no sample going into the extraction step. And then 3 negative controls for the RT-PCR and library preparation. And so when we look at the coverage values, the coverage plots for each of these controls, we can start to infer various interesting things about what's actually happened. So I'm showing here good controls. There's a couple of less than good controls in the next slides. But the positive controls that we're using are both a twist synthetic control. So twist is a company that basically synthesizes the SARS genome and distributes it for use as a control. The coverage plot on the top here shows that we're getting even coverage across the twist control, except for 5 positions across the genome. And that's because the control is only synthesized in 5 kilobase fragments. They can't synthesize a whole 30 kilobase fragment that covers the entire genome. What we also run is a positive control from cultured SARS-CoV-2 RNA from ATCC. That's the plot here on the bottom. And again, you see now the absence of those gaps for the twist control, but you also see that the coverage is more, that there's variability across the whole genome as well. That is related to the concentration of the SARS-CoV-2 RNA. On the right here, there are several negative controls. Sorry, I have to move my little window here. The top 2 are extraction negatives. So like I mentioned, these would be viral transport media going through the entire process. These generally want to be quite clean. And these little spots of coverage are random reads that map to the viral genome at levels of 10 to 100 reads. This is just for comparison. These runs are from batches that are sequenced to, on average, 1 to 10,000 reads per real sample. So we're talking about a sub 1% contamination level for the NTCs here. Similarly, these 3 on the right here are library preparation negatives, 3 duplicate water samples that go into the RT-PCR and library prep. Those should also be clean for the most part, with some trace coverage. We also run a SARS-CoV-2 negative HeLa control, just to account for any odd off-target effects on a genome that's more or less human. So this is kind of an ideal scenario where we would have these samples run alongside up to about 90 real samples through the process. But I do want to also share that coverage plots are a very overlooked source of information. So earlier on, I mentioned that we kind of try to reduce everything into a report with a heat map and a phylogeny, some information about each sample. Individual plots for 96 well plates are available, but we generally don't disseminate that downstream of our group. But in looking at them, you can actually start to see various details about each plate that goes through. So here on the left are 2 coverage plots from previously known positive samples that we did for one study. And on the top is a CT of 18 sample, which is a very concentrated sample. And the bottom one is a CT of 28. So that's about 2 to the 10th more dilute on the bottom. And you can see that, so this is just 2 points, but basically the trend holds where the more dilute a sample is, the more the coverage varies. And so you can actually see that whether there are deviations from the expected coverage profiles for high and low CT samples, and potentially communicate upstream and ask whether there are any sample storage issues. So for example, a low CT sample with 18 that looks like a CT 28 sample seems to imply that there is some degradation somewhere in the sample handling chain. Similarly, this is actually some information from one run where we started to see that we had reagent aliquot problems. You can actually have successful sequencing reaction with odd coverage profiles and gaps around certain areas of amplicons. This is usually due to either a temperature change in a PCR head or something happening with reagents. The third type of issue that you can flag with a coverage plot is a sawtooth coverage, which is basically sequencing of every alternate amplicon in the panel. This usually indicates that you've had an issue with your PCR subpool. So in this particular sample was a one-off where all of the samples on the plate generally looked okay. And then we just had some sort of issue with adding polymerase, let's say, to one sample in one of the subpools that just didn't generate the odd numbered amplicons, and they didn't get into library prep. The other thing that you can actually spot from coverage plots is that the negative controls do tend to have some random amplicon coverage. This has been reported, I think, by every site using the Arctic panel. Amplicon coverage looks like a broad peak, about 300 bases wide. These can indicate random contamination events in the laboratory or process deviations. So in this case, this is actually not, this is contamination, it's random contamination, but the fact that the sequencing is so high, we actually went back and saw that the process had changed, and we were actually sequencing these to a lot deeper coverage than we would have normally, leading to more signal occurring in the NTCs and more reads per amplicon. So we're trying to diagnose things like that with these coverage plots. Okay, going into kind of the last section. So we had, like I mentioned, we had this test established for most of 2020, and in the fall we had to evaluate some further optimizations because Illumina had released their COVID-seq test for research use only in Canada. It's very similar to Arctic in that it is based on the Arctic amplicons. And we kind of thought that it was a simple plug and play switch. There's a big benefit to it because it's a faster protocol with substantially the same reagents. But the main concern that we had was twofold. One was, even though that they had taken the same panel from Arctic v3, and they had told us that they modified some of the amplicons slightly for better performance. We had the same amount of amplicons. They also had additional human amplicons in their amplification panels, which would be used as positive controls for each sample in a diagnostic setting, but for our purposes, they really weren't relevant and would actually incur a large cost of changing the bioinformatics pipeline to adapt to a new panel. So we really wanted to see whether it was worth changing. And the way we went about that was first to compare just the raw coverage profiles between the two different panels. They're, like I said, they're substantially the same, but given that there are additional primers in the mix, we would expected some differences. We did here two figures that show side by side that yes, they generally follow a very similar trend in that there's more coverage in high amplicons, but they are different and they do perform differently. So based on this, we went a little bit deeper to evaluate what the impact of possibly switching to a COVID-seq panel would be on our research genomes. So we started to look at whether or not the quality of each individual library produced with the two panels, given the same input samples, is comparable. And so we prepared a bunch of libraries with real samples, sequenced to about 200,000 reads per library, and then calculated the proportion of each library that we're mapping to the SARS-CoV-2 genome. And so what we found was that the library yields were comparable below a CT of 28. In fact, the higher dots here, the green and blue dots, are the COVID-seq panel. It seemed to have a higher viral map rate in the sub CT28 samples. And then the red dots here are the Arctic v3 panel alone. So this is the viral panel with no human amplicons. That also seemed to have a consistent percentage of viral sequences in each library, until you went to more dilute samples beyond CT of 30. And the COVID-seq panel seemed to just not map as much to the viral genome. Now, this doesn't actually mean that the panel was inferior. What we started looking into it, but basically what started happening was that more of this panel was mapping to human. These are real samples. These weren't pure ATC samples. And so what you're seeing here is that the mixture of the viral amplicons and the human amplicons in the dilute viral sequences basically mean that it's starting to amplify the background human, which is exactly what the COVID-seq panel is supposed to be doing, but we didn't really need that data. What we were interested in is to just ask how good are the viral genomes? And so taking it another step further in examining the coverage uniformity, that is the variability of each amplicon across the viral genome, we could see that the uniformity was higher with the native V3 panel, probably because the additional human control amplicons in the COVID-seq panel were not interfering with the multiplex PCR reaction. And again, like I mentioned, the human amplicons were not really required for us, given that we knew that all these incoming samples coming to OICR were already positive for the virus. So the last bit of information that we had to look at before making a decision was basically, what do the viral genomes look like? And so the aim here is to generate nearly complete virus coverage, given that downstream researchers are going to be interested in looking at variants of concern and also relatedness between different samples and outbreaks, et cetera. We kind of took a philosophy of every base counts, and the higher coverage uniformity in the previous slide seemed to consistently translate to higher overall coverage of viral genomes, down to about the CT of 32 to 33 level. And so we're still basically getting a complete viral genome from the vast majority of samples that come our way. We do occasionally see samples in the sub CT 35 range, but those are kind of triaged upstream of us. And so based on results like this and some others internally, we basically made the decision to adopt the actual library prep workflow with the COVID-seq kit, but just swap out the primer set. And so what we're using continually in-house is the COVID-seq workflow, which is actually quite fast and robust to different steps in the laboratory, but with the upstream Arctic V3 primers upstream at the RT-PCR step without the human amplicons. So just to wrap this all up, just some perspectives from a genomics platform. I think in the last year, we've learned that rapid pivoting of cancer research assays to support viral genomics is possible. Kind of tricky. I think that one of the learnings from all of this is that different service products, whether it's for an outbreak investigation or upload to a public repository, do have different demands on the team. One thing I didn't show is that I think the record for one of our turnarounds between multiple departments at OICR to help resolve one outbreak was a little bit over 24 hours. And I think we had people handing off the samples around the clock, which is not something that we typically do, but this was a particular circumstance that everyone was happy to lend a hand. And we kind of gave a good answer to the partner that reached out to us. And lastly, with SARS-CoV-2 genomics, we pretty much realized kind of what we innately knew, that research use only sequencing processes can't just be adopted to a different genome, different samples. We really had to tailor and build around an assay around the types of incoming samples, and in our case, samples that are known positives with generally known quantifications, and then also the downstream needs. So like I mentioned, quick reports to just answer a phylogenetic question versus nearly complete coverage for other applications. And then also, being a data production platform, we always kind of joke and say that, we'll sequence as much as you want. But actually, some of the results that we've learned from our foray into COVID genomics is that more data and more information at the sequencing level is not always better, and actually can complicate a lot of times that you run into changes in the process that the additional data seems to flag at problems. So in conclusion, I just want to acknowledge the whole team in the Genome Research Platform. We're situated here at Mars. It's actually been extremely quiet over the last year, so it's been nice, but I think myself, along with many other people, are looking forward to the day where we can all sit downstairs and have a coffee without masks and move on. Thanks.